Heteroresistance is an interesting bacterial phenomenon when you talk about antibiotics that has been well known for many years. However, we're only really starting to understand the mechanistic basis of this phenomenon now, thanks to really pioneer research that has been published very recently. Indeed, the manner how bacteria become and respond to antibiotics is complex. And there are these phenomena called heteroresistance, persistence, tolerance that may be overlapping. And we are going to try to discuss a little bit more about it. Furthermore, heteroresistance seems to be a real phenomenon in clinical practice that may affect patients' outcomes. And this is very important because that will give us more tools to understand how to deploy appropriately antibiotics. Today, we have experts on the field discussing about this phenomena, and I want to welcome you to Editors in Conversation with our podcast, Heteroresistance, Mechanisms, Diagnosis, and Clinical Implications. The objectives of today's podcast is to understand the difference between heteroresistance, persistence, and tolerance, and not only difference, maybe similarities. Discuss on mechanistic basis in both in gram-positive and gram-negative bacteria, and elaborate on clinical implications and diagnosis of this phenomenon in clinical practice. This episode is brought to you by Antimicrobial Agents and Chemotherapy Journal, which is available at asm.org/AAC. I'm your host, Cesar Arias. I serve as the editor-in-chief of AAC, and this podcast is supported by the American Society for Microbiology, which publishes AAC. In fact, if you publish in AAC, you're a member of ASM, you can get up to 50% off on publishing fees. Visit asm.org/membership to sign up. And don't forget to check our latest issue of AAC with outstanding papers on mechanism, therapeutics, clinical pharmacokinetics, and clinical studies, among others. We also have a very nice collections of specific papers, particularly challenging clinical cases. Now we have a new section on antimicrobial stewardship and the new perspective on antimicrobial agents that is now published in more mini reviews in specific agents that will be very informative with leaders in the field. Well, join us to discuss this important topic, two experts. One is David Weiss, who is professor of medicine and director of the Center for Antimicrobial Resistance at Emory University School of Medicine in Atlanta, Georgia. And Dr. William Miller, who is assistant professor of medicine at Houston Methodist Hospital and Houston Methodist Research Institute and Weill Cornell Medical College in New York. Welcome, gentlemen, to the show. And thank you for taking the time to discuss this. Okay. So let's start from the basics. Our audience is very, different and diverse. So, David, let's start with definition. What is heteroresistance?
Yeah. Well, first, Cesar, thanks so much for the invitation. It's really a pleasure to be here. Heteroresistance is this kind of quirky phenomenon. I think we normally think about resistance, susceptibility, in that all the cells in a bacterial population will behave the same. So for a given antibiotic, that either 100% of the cells will be susceptible and be killed, or will be resistant and survive in the face of that antibiotic. But heteroresistance is a little bit different, where you see that a subpopulation only of the cells are resistant. And this can be an incredibly small subpopulation, so even 1 in a million, perhaps. When confronted with a given drug that a strain is heteroresistant to, as the susceptible cells are killed, those resistant cells can hang on, and over time they can replicate and increase to higher numbers. And so it is a way that the bacteria, some of the cells, can be resilient in the face of these antibiotics.
Yeah, so we are learning how in a bacterial population there are really different kind of citizens there and how they respond to drugs. And I remember the studies long time ago in the late 70s, early 80s about this phenomenon in Staph aureus. First with methicillin, for example, methicillin resistant Staph aureus, and of course when this vancomycin resistant or intermediate resistant subpopulation in Staph aureus emerged. Um, Studies that were initially published from Japan, Dr. Hiramatsu and others, that there was clearly a phenomenon, and that's where this population or subpopulation analysis came to along. But mechanistically, has been hard to understand. So, Will, why there's also this phenomenon called persistence that people even use in titles of papers to try to understand more or target certain readers, or tolerance, which is not quite resistance. So, but they may have sort of important clinical implications. So, do we know what the difference are between heteroresistance, tolerance, and maybe persistence?
So thanks again for inviting me to come on. And I think kind of the way to answer that question simply is to say that, Dr. Weiss used a really important word, grow, in the presence of the antibiotic. And so when you start talking about things like tolerance and persistence, you have bacteria for which you have decreased metabolic activity. And so these are bacteria that are in a dormant state or maybe not actively replicating in the presence of the antibiotic. But the rate of killing of these particular bacteria is much lower than you would see for a susceptible bacterial population. And so you can kind of think about that as we talk about, what we are talking about with a minimum inhibitory concentration versus what we talk about with, say, like a killing assay where we're looking at a bactericidal concentration and we're seeing a certain threshold drop in the number of growing cells in the population. And so, where the minimum inhibitory concentration is basically where we're not going to be seeing cell growth, a bactericidal concentration has been defined as, like a 3 log drop or a 99.9% killing. And so for tolerant cells, the time that it takes to kill the number of cells to get down to that threshold is extended as opposed to cells that we would think of as normally susceptible. So you get a real quick drop, you get a quick killing versus a much slower. And persistence is where you have differences in the rates of killing between the populations. So you're going to kill a certain population very quickly, and then you're going to have a lower number that tend to hang on for a longer time period. And so really it's going to be these metabolically quiescent cells that aren't going to be killed that then can maybe outlast the exposure to the antibiotic and then begin to grow again once they receive some sort of trigger or once the antimicrobial has dropped below a certain threshold concentration.
Well, let me put it in simple words, and I'm going to ask David to see if he agrees. So heteroresistant is based on a number of population of cells that may survive the antibiotics, whatever mechanism that is. Tolerance is that a population that can survive increasing concentration of the antibiotic, but you eventually, if you get to that point, you can kill them. And maybe with the heteroresistant, you can also potentially kill them if you get to a concentration of antibiotics. But the persisters persist, and the redundancy is that, and you will have very hard time killing them despite vast amount of antibiotics. What do you think, David? Would that be accurate to describe?
Yeah, I mean, I think the biggest difference there is right for the persisters, right? When the drug is present, they will not be growing, replicating whatsoever. So just right off the bat, there's the idea that persisters may cause a recurrence of infection, but they cannot cause like acute treatment failure, as you can see with the resistant populations of cells with the heteroresistance. And then I think one other thing that's really important to bring up as sort of part of the definition of heteroresistance is that the resistance phenotype is unstable in a sense. And so this is nuanced. So I want to make sure that I don't make a confusing mess out of this. But so the frequency of the resistant cells can change in different environments. If you expose to antibiotics, the frequency of the resistant cells can increase. However, after antibiotic exposure, without drug present, in heteroresistance, the frequency of the resistant population will retreat. It'll start to go down again. And so that difference there is characteristic of heteroresistance. If you enriched for a resistant cell and they stayed resistant no matter if drug is there or not, we would look at that as, hey, you may have evolved stable resistance, like more classical canonical resistance. So that ability to be flexible and change the frequency of the resistant cell population is really a critical hallmark of heteroresistance.
Fantastic. So instability, another part for the word. And in practical terms, we've seen this, although not recognized it, when the first e-test came to be used in clinical terms. And I remember particularly with the vancomycin story and Staph aureus, where you started seeing colonies inside that halo inhibition. So these are different subpopulations, and you start to just scratch your head. What are those? This is an aha moment. Okay, these are different cells, and interesting. So David, what are the in vivo consequences of this? You just mentioned something very important, which is depending on where the cells grow. And growing in an MIC, in a lab, in a very sort of regulated, strict medium is very different than growing in the blood or in a tissue. And the opportunities to have this flexibility increases. So what are the consequences of this heteroresistant in vivo?
Yeah, great question. Let me just say one thing to make sure to clarify. When I say that instability is a part of the definition of heteroresistance, this does not mean that you go to the freezer and your isolate, like one day is heteroresistant, the next day it's not heteroresistant. So heteroresistance is a reproducible phenotype of a given isolate. It's just that the frequency of the resistant cells in different environments can change. So as far as the in vivo relevance, so great question. At the beginning of studying this, we thought, like, how is it possible? If you have 1 out of 100,000 cells is resistant, how could that possibly lead to any real in vivo relevance, right? Let's say a mouse model at first. But what we found was, in fact, if those resistant cells can hang on and replicate, and over time, they can cause a complete treatment failure in these in vivo model systems, so that this can be extremely relevant in vivo. And there are really interesting aspects where, in some cases, even the host can impact what's going on and the selection, the enrichment of the resistant cells. So you can imagine that in the clinic, if you have this small population of resistant cells in a person, that could grow when the antibiotic is prescribed, and over time cause a treatment failure.
Yeah. So, Dr. Miller, you just published a paper in AAC about heteroresistant with this new CD4 antibiotic called cefiderocol. And you are trying to look about clinical impact. What can you tell us about this?
So, this came out of a particular case that we had published a few years before for a patient that had exposure to several different antibiotics, and then a relapse of a bloodstream infection that then was set up for cefiderocol treatment. And kind of going back to the population and looking, it was interesting because we had found that at a low level in the population, there was a certain number of bacteria that had a loss of a response regulator, which is a gene that controls the expression of other genes. And essentially what that did was decrease the expression of the particular pores that actively transport cefiderocol inside of the cell. And so what we noted in the population was that across these different cells, there was a differential susceptibility to cefiderocol, where some were killed quite quickly, and then there was a subpopulation where there was survival out to and beyond the breakpoint that we would normally think of for susceptibility for the antibiotic. And essentially, this, in complex with potentially some mutations in the intrinsic cephalosporinase that Pseudomonas has, the AMP-C gene, led to the potential where different members of the population had different susceptibilities to the drug. So some were high expressors of the AMP-C, had this mutation, had decreased import of the drug, and thus were able to survive at a higher population, at a higher concentration of the drug than the rest of the population. And so essentially, in treating a patient like that with the drug, if you have a high burden of infection, if you have an uncontrolled focus, the concern is then that those cells will persist, and that then you will have selecting a larger and larger proportion because you have that selective pressure there. You're going to select those out. And this is really an adaptive mechanism, and it almost at the population scale, where they can have a series of mutations that may have a fitness cost or a growth cost. And it's maintained in a smaller proportion of the population that they can then expand once the selective pressure is applied. And so this allows the bacteria to be able to kind of respond to its environment and potentially lead to potential treatment failures or relapses of the infection.
Yeah, and there are some emerging data that suggest that this could be linked to clinical outcomes. And David, you have a very nice paper about that phenomenon in fungi, where there is clearly correlation with clinical outcomes. So Dr. Miller used this word bacterial fitness. So what does it do to the bacterial fitness, this phenomenon? What is this population able to do when they develop this sort of less or more ability to resist antibiotics?
Yeah, so I think the fitness is a really interesting aspect of this. So that can, as the bacteria are able to resist antibiotics, they may lose the ability to grow as fast or other things that they need to do in the setting of an infection. And this actually dovetails with this aspect of heteroresistance, which is the instability. So it's actually the fitness cost of the resistant cells, the reduced or the increased fitness cost in the resistant cells that leads to the instability. So if the resistant cells were just as fit as the susceptible cells, once you expose to antibiotic and you enrich the resistant population, they would just remain that way because there would be no pressure to revert back to a predominantly susceptible population. But usually what it looks like, what we see is that those resistant cells have some kind of increased fitness cost that can't do quite as well as the susceptibles. So as soon as you take the antibiotic away, the susceptible cells now start to be the ones who are sort of outperforming and replicating faster, and they start to take over the population, and that's what pushes back down the level of the resistant cells.
Oh, that's fascinating. Huh? Bacteria are incredible. So let's talk about the mechanisms now, because this is so complex that you imagine the bacterial adaptation mechanism has to be very complex. Let's think about first the gram-positives. And I remember a long time ago, we published a paper in the anthracoccide, vancomycin-resistant anthracoccide, when there were subpopulations. This is a patient where suddenly the phenotype of vancomycin-resistant changed to vancomycin susceptible. And of course, the clinicians responded to change the antibiotics to vancomycin susceptible. And they use vancomycin, and then vancomycin resistant emerged as well. And what happened is there was a population of organisms of cells that had this plasmid that encoded the VAN-A operon, whereas the majority did not have it. But of course, when you put the pressure, is that population that took over. Then you remove, as you, David, explained, the antibiotic, and then the cells became, again, vancomycin susceptible, decided to just lose the plasmid as a mean to save energy. Bacteria are not stupid, I always tell. They're very clever, and they will use the resources. Will, in Staph aureus, that visa phenotype, what do we know about mechanisms about heteroresistance in Staph aureus, for example?
Yeah, so I think kind of one of the big things that we see in a lot of gram-positive organisms is that the mechanism tends to go through signaling systems that control the cell wall stress or cell envelope stress response. And so specifically, kind of classically with the heterogeneous vancomycin intermediate susceptibility phenotype, or the H-VISA, what was typically seen was that you would have these populations, again, that would survive at intermediate, but it's a smaller proportion. And if you look at the mutations, they tend to arise in the operons, so the VRA or the GRA, so VRA and GRA, and or the RNA polymerase or RPOB gene that change the way that the bacterial cell signaling is responding to the stress. So you get changes in the cell envelope, you get changes in cell wall thickness, you get differences in the charge distribution. And so this allows parts of the population, because that does to impact part of the growth, allows them to change that while the whole population, is growing. And so typically that's what we've seen, is that you'll get these changes, you'll get these envelope changes, and then you'll have part of the population that ends up as resistant to the antibiotic. And interesting, those pathways can actually have cross resistance. One with daptomycin, so some of those pathways will also impact your susceptibility to daptomycin. And some of the earlier work showed that some of these H-VISA strains will actually also have subpopulations that survive in the presence of daptomycin. And kind of where this may have come about from in the first place with staph is that things like daptomycin are very much related to antimicrobial peptides that the innate immune system makes. And probably we'll hear from Dr. Weiss with colistin as well. But these particular peptides will actually start engaging the signaling systems the same as we would see with Vanco or Dapto. And so we see that the same selective pressures may be induced by these innate immune peptides. And so this may be kind of underlying some of the phenotype of why we see some of these changes that are there and established in some of these organisms. More rarely, there have been some descriptions of changes, as you were saying. So expansion of the number of copies of, say, the vancomycin resistance genes in the van operon in plasmids, or potentially in the regulation of the expression of the van genes. That can also lead to this kind of heterogeneous population and this intermittent phenotype that you see. So really, it's kind of going to be the variable expression of those genes, either because of the number of copies that you see there, or because of the differential intrinsic regulation of the stress response pathways that the bacteria possess.
So that's a key factor, right? Particularly the cell envelope stress responses, and a very coordinated switching on of certain genes through 2 or 3 component regulatory systems that gram-positive bacteria have very specifically responding to this. And the mimicking with antimicrobial peptides is very important, because you think about that by the time you treat one of these organisms in vivo in a patient, they have already had to go through all of this, and all these systems are already on. So no wonder we sort of miss this. If we don't use the right drug at the right time or the right concentration, we're going to push this phenomenon to occur and eventually lead to treatment failure. So the gram negatives are quite different, because the cell envelope is very different. So you've been studying this, and we're publishing fascinating work on this, David. Tell us a little bit about that. We don't have time to hear the whole story.
Right.
But what are the more interesting mechanisms that you've witnessed in gram-negative bacteria of heteroresistance?
Yeah, thank you so much. So I think, the first one is the one that people have seen most often, is that you have gene amplification, which is, I would say for the clearest case is, for example, a beta-lactamase, which provides resistance to beta-lactam. So this enzyme can go and chop up the beta-lactam drugs. And so what happens is, if you have more copies of the gene encoding the beta-lactamase, you can have more beta-lactamase produced, and the cells that have an increased copy number will be more resistant. So how on earth does this happen? Because in grad school, definitely nobody was teaching me anything like this. And so this, what we see, and many people in the field have seen this, is that you'll have regions that encode the beta lactamase, and some distance, it can be maybe 2 kb, it can be sometimes 50 kb or more, which is just incredible. That you'll have repeat regions on the ends of this, and during the process of homologous recombination, you can have an increase in the copy number. And it's incredible also the number of copies that can sometimes occur can even reach into hundreds in some cases. And that the entire size of the genome of the bacterium in some cells can increase. Don Anderson published in a Klebsiella that the genome size tripled just from the unbelievable number of copies of a certain region encoding a beta lactamase that were reached in this strain. So
David, sorry to interrupt. Just to mention that in our hospital, that mechanism is one of the most common mechanisms of carbapenem resistant. And when they jump, they also actually knock out porins. So you have a gene amplification and the porins, and then that's it. And there's no carbapenemase genes that you can detect.
Yeah. It's amazing how flexible the bacteria are. And I would say, the question about what are the mechanisms, I would say there's heterogeneity in the heteroresistance, right? So the more we look, the more different mechanisms that people are finding, which is both exciting and scary at the same time. So I think the nice thing in terms of looking for it is you can see this mechanism with the amplification through genome sequencing. Although you may have to grow the bacteria in the presence of the drug and then compare to the sequence in the absence of the drug to see the difference in copy number. But I also want to highlight there for sure are mechanisms like, as Will nicely explained, that are totally independent of changes in copy number, right? So for colistin resistance, we see that this is more a signaling difference, that there's like a switch that gets triggered in some cells and not the other cells. And when we look actively to try to find amplifications, like we're not finding amplifications that have anything to do with colistin resistance in the strains that we looked at. However, other people have seen in some colistin heteroresistance, there can be amplifications, right? So this kind of highlights the heterogeneity in mechanisms, even among different isolates of the same species or different types of bacteria.
So, wow. So then as an ID physician, I wonder how I'm going to detect this and how I'm going to predict this. So let's talk about diagnosis, and I want to ask you, both of you, and I know, we have now more ability to sequence things, probably not yet quite there in real time, but I don't know if that is going to be enough. So what are your thoughts about detecting this in the clinical lab? Will, tell us some thoughts.
So it's going to depend somewhat when you're dealing with your isolate. One, about the mechanism, about the pressure that it's been exposed to, and then really, about how big is that population. So if you think about doing a standard antimicrobial susceptibility test, when we think about doing an MIC, we put in an inoculum of around 5 times 10 to the 5th. So if we're talking about that 1 in a million cell, number one, you'd have to pick that 1 in a million out and have it go in your inoculum. And 2, you'd have to hold it long enough to make sure that the few cells that are there are going to be able to grow out so that you could read it out through a visual test and see is there turbidity that's growing there. So I think for ones where it's a significant portion of the population, these are kind of trivial because those eventually are going to grow and they'll probably be caught by your traditional testing because they're going to make up enough that they're going to grow. But what we're worried about is those ones that are running under smaller amounts of the population, where your traditional antimicrobial susceptibility testing is just not going to be able to pick that up. And there have been several different things that, people have kind of tried to start to do to see. And one is kind of thinking about like, okay, can we use a bigger inoculum up front so that we can try to catch some of that smaller subpopulation and identify it? And then put it out on a lawn, and as you're talking about before with, gradient diffusion strips or maybe like a Kirby Bauer disc. Do you see where you have kind of these inner zones or these speckling of colonies that show, okay, we see some growth there. We see that there may be a population that's different from the overall susceptible population. The problem is that even those are going to be limited in the how low they can detect. So what is the smallest number of colonies. And so I think it's really getting down into those when we start to say, 1 in 100,000, 1 in a million colonies, where we're worried about those particular ones in a patient where you have maybe an uncontrolled source or a high burden inoculum infection where when you apply the drug, you're going to have enough there that you're going to have growth and escape. And so I think it's really that particular situation where, one, we have to define the clinical relevance to know for these different ones, and it may differ from bug to drug combination too, as to what that threshold is. And then two, so how do we identify that small number in the population to be able to call those strains heteroresistant, or at least give the clinician some sort of upfront knowledge that maybe this is something they need to be concerned about.
Yeah, that's going to be tough, I think. Any of these measures is going to be tough. David, any thoughts about how to detect it? Let's say we have the technology. What would you do?
Yeah, good question. I mean, I don't think there's any way around, other than you mentioned, Cesar, the possibility of genetics and genome sequencing. But if you want to measure something phenotypically, as Will just said really nicely, you have to have more cells than the frequency of the resistance in that population. There's no way around that, right? So especially for some of these newer approaches that are being developed for susceptibility testing that use a really small inoculum, like they will not be able to detect this. I do think that genome sequencing may, as it becomes faster, cheaper, it may have some role in this, because there are people working on this and some instances where certain genetic signatures have been linked to heteroresistance. However, I think that definitely will never capture all of it, in part because the bacteria are evolving all the time and changing, and you won't always know what genetic signature to look for. So I think in terms of what would we do if I guess the issue is, one, you have to start off with enough cells, as we said, and that can be a problem, right? Because you take a sample from a patient, and if you have to expand in any way the numbers of the cells, right, that is using time for the bacteria to naturally replicate, right? And we, of course, want the answer of the susceptibility as fast as possible, so you make the best decision in treating the patient. So essentially, we need an approach. I don't know, we don't normally think about speeding up the growth of bacteria, but I mean, it's not the worst idea that if we had a way to in vitro increase, get them growing as fast as possible, could we, get to that testing faster? But if you can get enough that come out, right, from the patient, then you want to see whether it be through a marker detection, some detection assay, or just phenotypically that they're replicating. See, in as few cells as possible, you can capture that replication event, that you can see that survival or replication with as few cells as possible.
So fascinating. I want to spend a few minutes thinking about this clinical implication of this. And I always say, I think resistance is unavoidable. Okay, so I wish everyone will think about that. That once you put a molecule like an antibiotic in a living thing like a human full of bacteria, it is going to happen. So instead of fighting resistance, we should accept it, right? And maybe think about how we mitigate it in the best way possible, And those are things like, you can do. Um, and one specific strategy would be to try to hit bacteria in different points, right? So that's the rationale for maybe combination therapy from the beginning. And there's some in vitro data, very nice evolutionary works that have been done to do this. So what are your thoughts about Weill? Should we use, and this could be very controversial, but I think we are in this safest space here. And I'm going to propose, maybe we should think about that. We maybe at the beginning, when you start treating an infection, of course you want to decrease the bacterial load, so drain the abscess, decrease bacterial load. We know that for a fact, right? But maybe you should just be very hard. Maybe we should try to do combination therapy from the beginning. And you don't need to continue for a long time, but decrease the number of cells low enough to give them more trouble to bacteria to have that step. They're going to have it, but then you do it. What do you think about it? Is it too crazy?
So I think this is where the understanding mechanism helps, because that helps us to try to come up with a rational combination therapy rather than just picking two drugs and putting them together to say, well, we're going to use two because we're worried about it being resistant to one or the other. And I think that really kind of goes back into, when you look at like gram positives and seeing that there's a synergy between membrane active antibiotic like daptomycin and then beta lactams, because the types of changes that are being induced to drive one phenotype may open it up to attack or being susceptible by another route. And so I think when we're talking about doing that, we should try to rationally pair together combination therapy to take advantage of a strategy that would really eliminate, the two different avenues that they would have toward going toward resistance. So, if we have gram negatives and we have gene amplification, and we're thinking that, the issue is going to be that we're going to have multiple copies of a beta lactamase gene and that potentially it's going to be jumping into a porin, then I want to say, okay, I want to try to pick an antibiotic maybe that's not going to be impacted by whichever porin loss that I'm going to guess that they have. So, if I know that it's going to be jumping into something like in Klebsiella, OMPK 35, 36, and I know that's going to be knocking down carbapenems, but maybe we'll have the option to get, something that's like a switteronic cefepime to get in and say, okay, I'm going to use that as my beta lactam. And then I'm going to say, okay, what's my enzyme? What inhibitor can I use to try to inhibit that enzyme to give my other drug the best chance of having activity? And so I think trying to pair those types of things together to really try to put your most active therapy on board is what we're going to need to be doing. And so you're going to have to really kind of understand how your bug is going to be potentially heteroresistant or, resistant to your antibiotics to be able to select that. And then understanding, like you said, it's resistance is inevitable. Well, persistence is going to be inevitable, right? Because we see that in susceptible cells. And so no matter what kind of combination or active therapy we're going to have, you're going to have some that are going to be in this kind of quiescent state. And so then I think, that, again, that's, about going back to the basics of ID of, source control and understanding our host. And so I think it's important that, we don't lose those aspects of things as well. Even though I know some of those sometimes can be a challenge to
yeah. So
in certain
so yeah. So that's going to require like a total different specialty, right, David? How do you teach this to clinician, infectious disease physicians, which is complex, and nobody has the grasp that you have on these mechanistic insights? What are your thoughts?
Yeah, I mean, I think both of you hit like great points in terms of the possibility of combinations and hitting the bacteria from as many different points. So I would say just one thing also is there's this strategy that we found, which says that for each strain, that it's not necessarily the property of a certain combination that it will work against a certain type of bacteria, like Klebsiella. But it actually matters like which Klebsiella and what resistance it has or heteroresistance. And we found that combinations that to where a strain is resistant to both of the different drugs, 100% of the cells are resistant to drug A and B, and you combine them, that it's very, very rare in our hands that you would see effective killing. But what we saw instead was that if a strain had been classified in the clinic as resistant to both drugs, but it was actually heteroresistant to both, then you could get this combinatorial effect of drug A kills the 999 out of 1,000, let's say, that are susceptible to drug A, and then you get the same thing to drug B, for example, and now you're knocking down to only maybe 1 in 10 million or less cells are surviving the combination, and hopefully there's a little bit of an immune system to help you out, and then the patient can get better. So that what Will said about these rational combinations, and that this is something that we could test for and then help use in a rational way, and which again highlights the need, I would say, for susceptibility testing capable of detecting heteroresistance as well as other phenomenon that we're talking about. So I think we really need that, and I envision this future where the antibiogram does not just say susceptible, intermediate, resistant, but also includes heteroresistant and other things as well.
Well, fantastic thoughts. Any concluding remarks, Will?
So, I mean, I think kind of partway, that's what our immune system has evolved to do, right? Attack from multiple different ways and work to clear those infections. And so I think that's why, despite these things having been there, I mean, the antibiotics are still remarkably effective, in a majority of the different cases. And so I think really what we're going to need to do is work on the diagnostic aspect. How do we identify these? And then that allows us, once we have a benchmark, to be able to come back to clinical outcomes and say, where does it matter? And then how do we rationally deploy, antibiotics and these strategies rather than just doing it indiscriminately? And so I think that kind of work to understand the mechanism, to be able to develop better diagnostics, all leads into how we then approach this from the clinical standpoint.
Well, beautiful. With that, I want to thank Dr. Miller and Dr. White with this phenomenal and very thought-provoking conversation. Thank you very much for attending our show. So there's much to do in the future, lots of exciting things to discover. We continue to follow your work very closely, so thank you for coming to the show. This is Cesar Arias, Editor-in-Chief of AAC, signing off.